Good morning, everybody. Yay, thank you so much for coming back and being ready to engage in our day two. I had such a great time yesterday. I thought all the talks were amazing, and then the energy and the interaction at the poster session, and apologies to the people I literally was having to kick out, quite fiercely. But that's a great sign. I'm glad that I have to do that because it meant people were really engaged. And so today we are going to have 2 more science sessions, so lots more science. And our at lunch is the optional workshop engaging the public on sensitive topics. And then we will have the panel for open platforms, AI and data sharing for multidimensional neurobiology. But first up, we have our first science session. So I'll turn it over to you.
Good morning, everyone. Can you hear me? Perfect. I'm Dennis Weingarten. I'm a postdoc in the Volume Institute at the Oregon Health and Science University, and I was very fortunate to be one of the Kavli Grass Fellows last year. And so this first session is about behavioral adaptations to changing environments, right? As we all have experienced in the last few weeks and months, sometimes changing environments are not that pleasant. And I'm very fortunate to introduce 4 speakers that will teach us different coping mechanisms we can learn from to maybe adapt to these changes. And so sometimes it feels like your brain is literally melting, right? And fortunately, there is adult-born neurogenesis. And so our first speaker of the session is Tracy Larson from the University of Virginia, and she will teach us that maybe we just need to grow a new brain.
All right. So first, I want to start out by thanking the Kavli Foundation and the Allen Frontiers Group for the invitation to present the work that my lab does. So the major question that drives my research program is what is the capacity for degeneration and regeneration, both within the individual and across species? So when we look across all animals, including vertebrates, we can find lots of examples of extensive regeneration, not only within the complexity of cells or tissues that are regenerated, but across the lifespan of these individual organisms. So for example, during metamorphosis of the butterfly from the caterpillar, we can see extensive remodeling of nearly all of the tissues, including the nervous system. Likewise, as larval fish transition to their adult form, they also remodel the majority of the tissues in the body, including the pigment cells that make up the adult pigment pattern. In the adult form, degeneration and regeneration can occur naturally and cyclically, and this is a process that we term adult plasticity. And perhaps one of the most striking examples in the natural world is the shedding and regrowth of antlers in the deer. And today I'm going to talk to you about another naturally cyclical form of degeneration and regeneration. During aging adult, regenerative processes tend to shut down, and this primarily happens through senescence, or the cessation of proliferation of progenitor and stem cells that give rise to these remodeling tissues. Of course, even so, there are lots of examples of organisms that prolong lifespan by delaying senescence and continuing to regenerate tissues, like that of the heart of the naked mole rat. And finally, all of us are aware here that all of these processes of degeneration and regeneration are contextualized by the developmental, the immediate, and even the ancestral environments of the organism. So all of these examples of remodeling and regeneration have a common set of features, and this includes degeneration or complete loss of a tissue with some component of cell death, and then regrowth of the tissue through the proliferation and survival of newborn cells that remodel that tissue. And so lots of amazing work has been done to identify the cellular and molecular mechanisms that drive some of these processes, and some of the examples I provided and several others. Critically though, at some point regeneration must stop. Otherwise, you can risk overgrowing the tissue and causing harm. Likewise, on the degeneration side, cell death must stop or risk irreparable damage to the tissue or even loss of that tissue completely. So this raises several really important questions that my lab focuses on. And those are specifically, what are the processes that initiate regeneration and degeneration? What are the mechanisms that limit the extent of degeneration and regeneration? And what mechanisms or processes stop regeneration and degeneration altogether? And a final really important question to ask in this context is, what mechanisms promote the maintenance of that tissue in its newly grown or regressed state. And it's really important to ask this because return to homeostasis is really important when you're thinking about a return to function of that tissue. So the primary system that we use in my lab is a white-crowned sparrow, a songbird. So on the breeding grounds, up here in Alaska, these are the wintering grounds of our bird, these males will sing their song to defend their territories and attract mates. And this is an example of a song from a Gambel's white-crowned sparrow, represented in what's called a sonogram. So time is on the x-axis, frequency on the y, and you can kind of read this like music. A typical white-crowned sparrow song is comprised of 5 syllables, a whistle, a warble, and 3 buzzes. So the neurobiological basis of singing behavior, both the behavior, the production of the song, but also the learning of the song, has been studied for over 50 years. And so we have a really good understanding of the neural circuits that are necessary for both learning and production of song. And that's represented here in this schematic. So the beak of the bird, this is the brain of a bird. The beak would be this direction, top of the head here. And there's this nucleus called HVC that underlies this ventricle and sends projections down to the robust nucleus of archipallium, or RA. RA sends projections to the hypoglossal nucleus, which then projects out to the muscles of the syrinx, and passage of the air through the syrinx results in the production of song. So particularly useful to my lab, both during transition from breeding to non-breeding seasons, this pathway undergoes dramatic morphological and physiological changes. So as birds transition into breeding season, day length increases. This increases gonadal production of testosterone, and that testosterone then prompts the growth or nearly doubling of volume of HCC and RA, and these changes altogether result in the production of high-quality song that's produced at a high rate. As birds transition into non-breeding seasons, the gonads stop producing testosterone, HVC and RA regress in volume, and the birds discontinue singing. So I want to point out that the growth and regression of HVC and RA is not equivalent. So the growth and degeneration of RA is due to actually expansion and retraction of the dendritic arbors and the soma size of individual neurons within the nucleus. Whereas in HVC, the growth is actually due to nearly a doubling of the neurons that send their projections down to RA, and then subsequent death of those neurons as the birds transition into non-breeding seasons. Okay, so now I'm going to briefly dive into some of the cellular interactions that drive this growth and degeneration. And throughout this, I'm going to give kind of an overview, and I'll present data alongside in case you really want to dig into some of the data. Okay, so just to orient you again, HCC underlies the VZ and sends projections down to RA. Within the VZ is where the neural stem cells and neuroprogenitor cells reside. These cells are continuously proliferating and sending migratory neuroblasts into HVC. It takes around 1 week for these cells to migrate in and start to differentiate. Over the course of the next week or so, they find their location to reside, and then over the course of the next 2 weeks, send their projections down to RA. In stable non-breeding conditions, the proliferation rate of these progenitor cells actually balances cell death of neurons, and so we maintain a stable number of neurons in HVC. As birds transition into breeding conditions, as I mentioned before, testosterone levels go up, and this actually drives an expansion of the vasculature. And these endothelial cells increase their production of VEGF and BDNF. Sorry, let me grab some water. And it's actually the production of these neurotrophins that support the addition of new neurons and account for the growth of HVC. As I said before, when the birds transition into non-breeding conditions, testosterone levels drop, and there's this massive wave of cell death that happens. So around 60,000 neurons in HVC die through caspase-mediated cell death within just 1 day. At the same time, coinciding with this death, we see that proliferation of the progenitor cells nearby actually increase in their proliferation. So one of the first questions that we had in the system was whether or not the cell death was dependent on the physiological state of the bird and the tissue, so the presence of testosterone and these neurotrophins, or if it was actually responsive to the cell death in HVC. And so what we did was we took birds through this regression transition paradigm and infused a cocktail of caspase inhibitors directly into HVC. This was showing that infusion. And what we found was that, of course, the cocktail of caspase inhibitors did prevent cell death, as we'd expect. But we also found that the cocktail of caspase inhibitors reduced proliferation rates in the VC. And so this indicated to us that the proliferation of these progenitor cells was responsive to the state of cell death as opposed to the physiological state of the organism. And so this led us to ask the question whether or not the same relationship existed during growth of HVC. And we're really interested in asking this question because you could imagine, I mentioned there was this increased survival, and so you could imagine then that would downregulate proliferation in the VZ and in total limit the total growth of HVC. And so I'm actually not going to talk about those experiments today, but we do have several experiments ongoing to address that question. The other kind of what I'm going to talk about today is thinking about whether there are natural examples of this kind of variation in degree of regeneration and taking a more comparative approach. Okay, and so in fact, we do see variation in the white-crowned sparrows in terms of the degree of plasticity that they can undergo. And so I mentioned at the beginning, we work on the subspecies Gambelli of white-crowned sparrow, and they're highly migratory. There are actually 4 other subspecies that have varying degrees of migratory behavior, including Nutali, or the Nuttall's white-crowned sparrow, that's fully resident around the area of Berkeley, California. And there are a couple examples, a couple reports that suggest that plasticity in Nutali is reduced compared to that in Gambelli. And so this and some other kind of anecdotal reports has led the field to kind of make this assumption for a long time that the more migratory a songbird is, the more plastic its brain is. And so in my lab, we get really excited about these kind of what-if questions. And so we wanted to ask, okay, what if this is actually generally true, that the more migratory an animal is, in this case birds, does that actually drive a variation in the degree of plasticity? And so we began thinking about possible mechanisms. And kind of one hypothesis that we came to, which is rooted in the finding that HVC cell death leads to proliferation, was that perhaps the act of migration actually increases cell death in this region, and that stimulates proliferation. And we could imagine that that increase in cell death could be due to things like hypoxia or glucose availability, a decrease in glucose availability, alterations in melatonin signaling, as these birds are actively migrating. And so to test this, our first pass, we accessed a historical data set of tissue that was collected from Gambelli as they migrated north and south, and also kind of same time points of the year from Nutali. And we just went into the tissue and by morphology counted cell death. And so here I have plotted cell death in Nutali, and you can see this across the time of year, and it's pretty flat line. So this suggests there's not seasonal variation in cell death in Nutali. Alternatively, when we look at Gambelli, we actually do see an uptick in cell death within HVC during their spring migration. And so then, coming back to what I had said about cell death then prompting proliferation, we could imagine that this might then drive an increase in proliferation that could drive the growth of HVC to be larger. The other thing to think about, though, is that to grow a nucleus, it's not just proliferation, right? We don't need just the cells coming in, we actually need to maintain those cells. And so we did a little literature deep dive and came across an extensive literature showing that exercise actually promotes the survival and increases in neurogenesis in lab-reared rodents. And in fact, that a lot of this, the effective exercise is mediated through an increase in production of BDNF, which is a neurotrophic factor that supports neural survival. The other thing that we found in our deep dive was that the serotonin reuptake inhibitor fluoxetine actually can bind to the receptor for BDNF called TrkB and can increase signaling of BDNF. And so what we decided to do for this, to test whether or not increases in BDNF could change plasticity in HVC, was we gave birds oral doses of SSRIs and did this for 14 days and then assayed singing behavior as well as some cytoarchitectural features. And so what we found was that our vehicle-treated birds, of course, don't sing in this experiment, but the birds that are treated with the fluoxetine actually do increase singing by around 7 to 10 days after the onset of treatment. You can also notice, though, that the song's not fantastic, and I can talk a little bit afterwards about why that might be. The other thing that we found is in HVC, this is showing neuron number, we see an increase in the density of HU-positive neurons within HVC. Of course, then we wanted to look at volume and say, okay, does HVC grow? And in fact, we find that it doesn't grow. But what we do find is that RA actually nearly doubles in volume very quickly. And in fact, this is much quicker than the timeline that we would see RA naturally grow either in the wild or under our standard lab conditions. And so what we think is happening is that in this case, the fluoxetine is kind of bypassing this upregulation by testosterone of the endothelial cells, and instead is stimulating growth of RA that then provides the support for where these new neurons will come in and project. Okay, so at this point, you're probably thinking, like, what does this have to do with climate change? One of the things that we've noticed in songbirds, and actually across lots of birds, is that they're spending their winters further north, so they're not migrating as far as temperatures warm. So this is just a map showing kind of the change in range. And so with some of the preliminary data that I've shown you, you could imagine then that this decrease in migratory distance might actually decrease neuroplasticity, could then result in a decrease in behavioral plasticity, and perhaps even a decrease in flexibility or adaptability of the species as a whole, especially in context of a subsequent environmental perturbation. And so just very quickly, this is a distribution map across the time of year. We actually think we've identified a shift in the wintering range of Gambelli. And so here at this arrow, you could see that there's this purple area here. They should be down in this area. And we have some reports from our field site out in eastern Washington that local residents actually do see these birds in the winter. And so this year we're planning to go out and collect in the winter and actually go in and assay kind of the degree of neuroplasticity in this subpopulation. And so to wrap up, I have a graphical representation of our hypothetical model. And I'm actually putting this up here not to force us to walk through this, but to make a couple points about the future and challenges moving forward. And so first, I'd like to advocate taking a little bit more of a holistic approach to thinking about mechanisms that are driving neuroplasticity by thinking about how complex cellular and molecular interactions are occurring across cell lineages, but also across the entire kind of lifespan of that tissue. Second, I think we also need to continue to incorporate analyses of these diverse cell types, not just neurons, and especially thinking about the cells that are in direct contact with the environment. The last thing I'm going to think kind of more broadly and advocate for kind of two more approaches. One thing that I think that we need to remember, and perhaps not in the coming couple of years, but ultimately, is that we need to think about kind of reintegrating the phenotype of the whole organism, right? So in order for a behavior to be useful for an organism, that has to occur, that behavior and the neural changes that drive that behavior need to happen in coordination with the whole physiology and morphology of that organism. And so down the road, we're going to need to think about how are these changes being driven, and I would propose probably by hormones, to coordinate that physiology, morphology, brain, all to have this behavioral response that's appropriate. And finally, the last point I like to make is thinking about scaling our observations and principles across levels of biological organization, right? So you can think about taking principles from landscape ecology and natural selection and asking if these principles could inform our understanding of how cells interact with each other in the tissue landscape, and how that might drive things like fate specification or the likelihood to die, the ability to compete for resources. And likewise, we can take our understanding of these complex cellular interactions in that tissue landscape and apply them at kind of the macro ecological level and ask questions or apply those principles to understand kind of more complex macro ecology systems. The last point I'm going to make, I realize I'm just like a minute over, is that actually we had a proposal in to actually address these two final points that I made. It was an initiative called ADAPT that was primarily funding postdoctoral research, collaborative postdoctoral research, and unfortunately we recently had the funding pulled for this. It was an internal grant support, and so when there was the big budget cuts that came through, they pulled the funding, but we're kind of still on pause for that initiative. So to complete this work, I have a fantastic group of trainees, including a very healthy group of undergrads who did a lot of this amazing work in our lab. I'd also like to thank past funding, including NIDCD, NIMH, the Washington Research Foundation. Most of our work currently is funded, or at least the work that I talked about today is funded by NIGMS with Amira. And then finally, I'd like to give a shout out to the Allen Frontiers Group and thank them for providing our funding for the Allen Discovery Center for Neurobiology and Changing Environments. Thank you. This is our field site.
Okay, as usual, trainees first. Please state your name. And let me start with a question.
Okay.
So, some of the subspecies migrate less. Do you think they still have the circuit intact, and it's just that their testosterone levels don't fluctuate as much? And if you could brute force artificially changing testosterone, would you see the same plasticity?
So they do have plasticity, yeah, so like Nuttles, for instance, they do actually have plasticity in the song circuit. It's just not as extreme as the Gimbelli. So when you go in and measure their non-breeding volume or cell number versus like relative to breeding, there's just not as much of a dramatic change. But they do have plasticity. Every temperate songbird that we've examined has plasticity in the circuit.
Okay.
Yes.
So Jan Siemens from Heidelberg University. I was wondering about the cell death you mentioned. If I understood correctly, you said 60,000 cells in one day.
Yeah.
And that is induced by the seasonal change and drop in testosterone. So how do you get this all or none type of response from something that I would assume is sort of gradually going down?
Yeah, so that's in laboratory conditions. I kind of glossed over how we do this in the lab. So in the lab, we actually will transition birds from short day photoperiods, or like 8 hours of light, to long day photoperiods, about 20 hours of light. Because these guys breed up in Alaska where it's almost like full light all day long. We also give a subcutaneous testosterone pellet because I think it's the quality of light is just not high enough to get their testosterone levels up to what we see in the wild. And so when we do that lab manipulation, the processes occur much faster than they do in the wild. Yep.
Hi, Tracy. Martin, amazing talk. That's great. Is there much known about when you have all those cells dying, how that gets cleared out, how that affects pH on that fluid, and
No,
like all the...
that's definitely something that we're really interested in, because in a mammalian system, if you had that much cell death occurring, there'd be likely lots of inflammation, debris kind of hanging around, and then gliosis, and then ultimately loss of the behavior. And one of the really cool things about our system is that, like, yes, this happens really fast. It's super clean. The other thing is that the birds are still capable of singing. And so even with that amount of degeneration, so losing half the circuit, they're still actually able to produce the behavior. And I think that's a really remarkable thing when you're thinking about degeneration, regeneration, is that they can still perform the function of that tissue. And we have lots of ideas about how they're keeping so clean. And one idea is that they're diversifying the modes of cell death. So when I said, this is mediated through activate caspase 3, there's actually a couple other modes of programmed cell death that actually engage that pathway. And we think they're diversifying the modes of cell death to kind of tampen down the total kind of inflammatory versus pro-neurogenic response. Sorry, that was like way more than what you were probably wanting to know.
Would you expect to see difference in transcytosis?
And sorry, in what?
In transcytosis, that's part of BBB clearance. Because if you see a lot of near kind of inflammation or decrease in depending on plasticity.
Yeah, so we did a study. Yeah, so we did take a look at inflammation, and we actually couldn't find this is the bird system, so it was very kind of not the best way to assay inflammation. So we just did qPCR for some of the client classic inflammatory markers, but we didn't see any any signal of inflammation. We do see that microglia become activated and take on that kind of responsive morphology. Again, we don't have great markers for microglia, so it's a little hard to know, are they in the M1 versus M2 state? Like, what's the response that they're driving? But yeah, I would expect to see some changes.
With that, thank you so much, Tracy.
Yeah, of course. Thank you, guys.